Okay, just allow me a short remark on this abstract. I would like to thank the organizers for selecting this abstract to be presented here, especially Rob, who knows how much in love I am with Australia, so I love to come here every time I can. This abstract is the work of Julia Krall-Pointner. She is a postdoc in my lab. I'm fading out of research, so to speak. I'm only on a part-time contract with university, and I'm currently heading a technology platform. But this work, all the credit goes to Julia. And actually, I wanted her to come here, but she has 2 small children, so she was reluctant to come. So I had a good excuse to come. Okay, so if you read the title in the program, you see that I added a question mark, and you will see in a couple of minutes, you will see why. Okay, oh, that was too fast. So I think I can go quickly over this background slide. We heard a lot about several plasminogen binding proteins already in this conference. Plasminogen RKT, that's the plasminogen receptor I will talk about, has been recently or not so recently discovered as a transmembrane receptor for plasminogen. Its overexpression leads to more plasminogen binding to the surface. Its knockdown or deletion leads to less plasminogen binding on the surface of the cells. It's widely expressed in various cells. Oops. I have here of the many functions which have been described now for plasminogen Plg RKT. I have listed a few which are related to cardiovascular disease or might have some relevance to cardiovascular disease. So plasminogen RK2 is expressed on activated platelets and is involved in local fibrinolysis. Nikki has shown that in a nice paper and published in Blood a couple of years ago. It's also expressed in adipose tissue, and it's important to prevent adipose tissue dysfunction and dysregulation of metabolism in these cells. It's modulating atherosclerotic, so the uptake of dead or dying cells, and it modulates also the polarization of macrophages from M1 to M2 phenotype. Oops. A couple of years ago, we published this paper in Blood. This was a PhD student, very talented one in my lab. And we got interested if plasminogen receptor K2 might be differently expressed on monocyte subsets. So maybe I should allude a little bit to monocyte subsets. In humans, we have 3 different monocyte subsets. The so-called classical ones, they are CD14 positive, CD16 negative. They make up 80 to 85% of the circulating monocytes in our blood. The intermediate ones, they are CD14 positive and CD16 slightly positive. They make up in a healthy person, 5 to 7 to 8% of the circulating monocytes. And the so-called non-classical ones, which are only slightly positive for CD14 and strongly positive for CD16, which make up the rest. And when analyzing these human monocytes, we found that the intermediate one, and I should say the intermediate one are the inflammatory monocytes, express the highest levels of the plasminogen receptor, protein-wise, and also on the level of mRNA. And interestingly, these intermediate ones also express the highest level of urokinase plasminogen activator receptor, urokinase receptor. When we looked at plasminogen binding in these cells, again in blue, the intermediate monocytes showed the highest level of plasminogen binding. The classical, the non-classical bound only minute amounts of plasminogen. This plasminogen could be competed off by EACA and by an antibody against the receptor. When we took these inflammatory intermediate monocytes and exposed them to a Boyden chamber assay to account for migration of these cells, when we added plasminogen to the system, we got an increase in migration in these intermediate monocytes. This was not affected by an isotope antibody. It was blocked by an antibody against the receptor. It was blocked by EACA. It was blocked by the amino terminal fragment of urokinase, and it was blocked by aproteinin. So these were the in vitro results. And then we went into the mouse system. We did this study in cooperation with Lindsay Miles from La Jolla, and she provided us with the mice, the plasminogen receptor knockout mice. In mice, monocytes are a little bit different. In mice, there are only two subsets of monocytes, so-called Ly6C high and Ly6C low. The Ly6C high are the inflammatory ones, the Ly6C low, the anti-inflammatory or not inflammatory ones. And we exposed our mice to a sterile peritonitis model using dioglycolate, and we used wild type and receptor knockout mice. Before exposure or injection of dioglycolate, we didn't find any monocytes in the peritoneum. We found a huge increase a couple of days after the injection. This was true in male and in female mice. And what was interesting was that the plasminogen receptor knockout mice, the recruitment was impaired. So it was reduced to half of the wild type. This was only true for the intermediate monocyte, or the Ly6C high, the inflammatory monocytes in mice. The Ly6C low, there was no difference in the recruitment. We also looked into macrophages, or the expression of Plg RKT on human macrophages. And as you know, macrophages come in many different flavors. So there are the naive macrophages, we call them here M0. Then there are the M1 phenotype, the inflammatory one, which you can polarize in vitro by using LPS and interferon gamma. And there's the M2 phenotype, the non-inflammatory, which can be polarized in vitro by IL-4 and IL-13. And again, the inflammatory macrophages, like the inflammatory monocytes, express the highest level of plasminogen receptor in protein and also in mRNA. Migration similar to what I showed you for the inflammatory monocytes. So the inflammatory macrophages migrated best or better as compared to the M0 and the M2 macrophages. And this here, you probably can't see it very well. You have to believe me, but these pictures here brought us to what I will present to you in 2 or 3 minutes. These were slices of human atherosclerotic plaques, and we stained them with a marker for inflammatory macrophages, CD8T, with plasminogen, and with an antibody against the plasminogen receptor, and we found clear colocalization of inflammatory macrophages, of the receptor, and of plasminogen in these are human atherosclerotic plaques. So, another evidence which pointed us into the direction of atherosclerosis and a possible role for these different monocyte subsets is listed here. These are cardiovascular diseases, cardiovascular pathologies, which are associated with increased counts of intermediate monocytes. So that's in acute myocardial infarction, in unstable angina, acute chronic heart failure, stroke, AAA, and interestingly, high levels of intermediate monocytes are independently predictive of cardiovascular events in randomly selected humans. The non-classical monocytes, they're also increased in chronic heart failure and in AAA. So this was another indication that maybe something was going on with the different expression of the receptor, the different binding of plasminogen to these different subsets of monocytes. So we hypothesized that monocytes and macrophages are critically involved in the development and progression of atherosclerosis. We have shown that this RKT receptor, the plasminogen RKT, modulates migration and recruitment of monocytes and macrophages. Inflammatory macrophages, the receptor and plasminogen co-localize in human atherosclerotic plaques. So we hypothesized that the receptor might be involved in the pathogenesis of atherosclerosis, and so we aim to study the effect of a knockout of the plasminogen receptor on atherosclerosis development. So what we did, we used LDL receptor knockout mice and LDL receptor plasminogen receptor double knockout mice. We fed them a high cholesterol diet for 16 weeks, and for 6 weeks we controlled for body weight, we controlled for lipids. I can tell you in advance, I will not show you the data, there was no effect on body weight, there was no effect on lipids. We looked at the histology of the aortic root lesions, and we did ENFAS of the whole aorta of these mice. And we, as I said, we exposed these mice for 16 weeks and for 6 weeks to this high-fat diet. And we think we were smart starting with the 16 weeks, because the longer exposure, the more atherosclerosis. If there is an effect, we should see it in the 16 weeks mice. To our disappointment and surprise, I will have to show you a couple of rather boring slides. Nothing happened in the 16-week mice. So the plaque area was the same in the LDL receptor knockout mice and in the double knockout. In the total aorta, in the arch of the aorta, in the thoracic aorta, in the abdominal aorta. Nothing happened. We did blood cell counts. Again, nothing happened. So this was not too surprising and too disappointing. We didn't expect that there would be a big difference. So leukocytes, neutrophils, monocytes, T cells, B cells were the same in both, in the blood, in both phenotypes, or genotypes, sorry. Plaque area, next disappointment. Plaque area, same size. Necrotic core, same size in the LDL receptor knockout mice and in the double knockout mice. Collagen content, elastin fiber breaks, again, no difference. So Julia got more and more frustrated. Macrophages, foam cells, same levels in the plaque. The only difference was a difference in T cells, so we don't know how to interpret that for now, but this was significant. So there was a reduction in T cells in the, not in the blood, but in the plaque. So you think you have a bright idea, you know, and then you're disappointed. And fortunately, we had 6 weeks of high-fat diet, and this was then really surprising. Now, in the 6 weeks, we had a clear reduction, a significant reduction in plaque size in the knockout mice, so in total plaque area in the entire aorta. It was significantly different in the arch. There was no difference in the thoracic aorta, but there was again a significant difference in the abdominal aorta, which is quite nice actually, because the flow is much more turbulent in the arch and in the abdominal aorta as compared to the thoracic aorta. So this might have something to do with what we have seen here. And the blood cells, again, no difference. Okay, so, but this was to be expected. And I got this slide just 4 hours ago or 5 hours ago. Julia sent it to me. And she was very excited because there was a significant difference in collagen content between the LDL receptor knockout and the LDL plasminogen receptor knockout, double knockout. So there was more collagen present if the plasminogen receptor has been knocked out. So finally, we got some interesting results. We are not sure what that all means, that you have an early onset of atherosclerosis, which is obviously dependent on the plasminogen receptor, and the longer you let the mice sit, the effect obviously disappears. So Julia has summarized that here. Long, 16 weeks, high-fat diet, nothing except for the reduction of T cells in the aortic valve plaques. 6 weeks, short-term exposure, reduced plaques in the aorta, elevated collagen levels. Okay? So what Julia is planning to do, she wants to do, or she is actually currently doing, so that's the true reason why she had to stay at home. No, I'm just kidding. She is analyzing the cellular composition of the plaques right now. She will look into plaque stability, necrotic, measure the necrotic core, the elastin levels. We will also do 12 weeks exposure to high-fat diet to see if we get some sort of a kinetic, a strong effect at the beginning, which fades out after 12 weeks and which disappears after 16 weeks. And we'll do a both-way bone marrow transplant, so transplant plasminogen receptor knockout bone marrow into wild-type mice and wild-type bone marrow into the plasminogen receptor knockout mice to really be sure that it's the innate immune cells doing the job. And with that, I say thank you to all, to you for listening and for being here despite the nice weather outside. Thank you very much.